biotechnology, it is made up of two words. One is technology, another one is bios. So it's very well known to you that what do you mean by bio? Bio means life or living organisms. Okay, so life or living organisms. And what do you mean by technology? Technology means application of the knowledge of science for the welfare of the humans, or for the welfare of the humans. Simply, we can say likewise. So here, technology in simple manner, it refers to application of the knowledge for welfare. So here, what we are doing, biotechnology, it's nothing but it's a branch of science which generally deals with it uses the living organisms for the welfare of the humans. So that is, in simple manner, we can say biotechnology. Okay, so we will have various applications of biotechnology, and in this we will use the living organisms. Like, let's say, sometimes microorganisms can be there, sometimes we can have the living components can be used, like genes can be used likewise. So that is generally nothing but the biotechnology. Okay, so here we'll be simply applying, we'll be using them, and we'll be using this for the sake of welfare of the humans. So that is one thing. So here, when we talk from the perspective of biotechnology, the word biotechnology has actually coined in the year 1919 by a scholar called Karoli Ericki. So is the scholar who coined the term biotechnology. Okay, so here there are so many types of biotechnology with us. So biotechnology can be with respect to the industry, so we will call it as industrial biotechnology. Biotechnology can be there with respect to health and medical field, so we will call that as, like, let's say red biotechnology. Or biotechnology can be there with respect to, we can say, agriculture or agricultural products, so we'll call it as green biotechnology. So based on the targeted things, or based on the field which we are picking, biotechnology can be of various types. So when we talk about biotechnology, there are some important methods of biotechnology. Okay, so one important method of biotechnology is known as genetic engineering. Genetic engineering is one important method. Okay, next important method, or other important method, is, in simple way we can mention it as chemical engineering. We'll call it as chemical engineering. Okay, so here the chemical engineering, also technically known as something called bioprocessing. So you remember this technical word called bioprocess. Bioprocess, in simple manner to say, bioprocess refers to chemical engineering. So what we will do in chemical engineering, so you might have heard that we'll produce a lot of vaccines, we'll produce a lot of hormones, we'll produce a lot of such kind of chemical products. Okay, either through the process of fermentation or either through the process of some engineering methods. Okay, so like production of some vaccines, production of some hormones, so certain chemicals. Okay, so this would be part and parcel of the subject matter of chemical engineering. So here what we will do, we will try to make sure that the conditions will be there in sterile manner, and under controlled manner we will try to develop certain microbes or certain particular things. Let's say, for example, we need to produce antibiotics. We need to produce antibiotics. So let's say, let's take any antibiotic, let's say, for example, ampicillin or amoxicillin, or let's say we are producing any antibiotic. So there we need particular bacterial growth. Okay, so by the help of that we can kill the toxins. So by the help of that we can kill particular bacteria. So under controlled manner in chemical engineering, they will try to produce the specific products required to produce that particular antibiotic. So that would be the subject matter, and that would be the work done under chemical engineering. Okay, so this is one thing. So this is general way, and this is one method of biotechnology. Okay, so other method and other important method is genetic engineering. Okay, so the word genetic engineering itself talks that we are playing with something called genes. Okay, so genetic engineering means it is nothing but, in simple manner, it is nothing but the process of genetic modification. Genetic modification, or in other words, in simple manner, we can say that it is a process which includes alteration of the genetic composition of an organism. Okay, every living organism has a specific genetic composition. Okay, so what we'll be doing in genetic engineering is that we will be modifying the genetic composition of that living organism, or in simple manner to say, we will be altering. So alteration of genetic composition of an organism so as to produce either new form of organism or better form of organism, or to remove certain defects in that particular organism. It can be for any purpose. Okay, so that is one thing. So here, when we are talking about the alteration of the genetic composition, so genetic composition means what? Okay, so we also have to understand what is that. Okay, so if we talk from the perspective of living organisms, so when we take a living organism, so every living organism is made up of various organ systems. Okay, and various organ systems are in turn made up of various organs. Okay, so various organs are again composed of different tissues. Okay, so different tissues are again made up of cells. So from this perspective, cell is the basic structural and functional unit of a living organism. Okay, so here, when we talk from the perspective of cell, cell is again made up of different components and compounds. Okay, so if we take, for example, so if we assume, or like let's say, if we take that this is a cell. Okay, and usually cells will consist of various cell organelles. Okay, it is well known to you that there would be various parts in it. So at the center of this cell, there we will have something called nucleus. Okay, so nucleus is nothing but, nucleus would be located at the center of the cell. So nucleus. So within the nucleus, we will have some thread-like structures. So if this is the nucleus, and if this is the nucleus, so then within the nucleus we will have some thread-like structures called chromosomes. Okay, so we are just depicting chromosomes, and chromosomes are the thread-like structures which are made up of something called DNA. Chromosomes are made up of DNA. Okay, and DNA in again it is made up of something called genes. Okay, here genes would be the packets which consist of information, that is genetic information. Like for example, now if you talk about this pen drive is nothing but a tool which consists of some information related to our class, let's say simply. So what we will do there, what we will do with the help of this pen drive, we will try to transfer the information, which is digital information which is there, from one device to another device, from one computer to another computer likewise. So in the similar fashion, what will be done? So genes will have this information that is stored with them, and DNA is the place where these genes would be located, and DNA would be again in turn, it would be present in chromosomes. Okay, so here we are saying that these chromosomes are DNA or genes, they are generally present in the nucleus. So most of the DNA or most of the genetic information would be generally present in the nucleus of the cell. Okay, so we will call that kind of DNA as nuclear DNA. Nuclear DNA. And small amount of DNA or genetic material will also be present in mitochondria. So mitochondria is another cell organelle which is also known as the powerhouse of the cell. So small amount of DNA will also be present in the mitochondria, which is another cell organ, which is known as the powerhouse of the cell. Okay, so in this fashion, certain portion of DNA would be there in nucleus, it is nuclear DNA, and some portion of the DNA would be present in the mitochondria, so that is known as mitochondrial DNA. Okay, so here when we talk from the perspective of this mitochondrial DNA, so an offspring will receive the mitochondrial DNA only from the mother. Only from mother. Okay, remember this. So if there is any mitochondrial defects, then the mother would be responsible for having that. So you just remember that mitochondrial DNA would be inherited only from the mother. So this thing also you have to categorically remember. Okay, is that clear? So now, so this is with respect to DNA. And DNA is again made up of, it will have its own composition that already you know, like DNA is made up of some things like nucleotides, it is a polynucleotide chain, all those things. Yes.
So, you mentioned about mitochondria, so it will be present outside nucleus, right?
Yes, yes, yes. Within the cell, we will have various cell organelles. So cell would be generally composed of some liquid material called cytoplasm. So various cell organelles would be floating in the cytoplasm. So there mitochondria would be one cell organelle likewise. So that is known as the powerhouse of the cell. So likewise, so this is how DNA and genes would be there. So now what we would be doing, these genes will contains the genetic information. Genetic information. Okay, so through the transfer of the genes, genetic information would be transferred, and by that living organisms will be made up. So here when we talk from the perspective of these genes, so genes are responsible for certain manifestation of certain characteristics in the individual. Okay, so from that perspective, when we talk from the perspective of genes, so genes, we have said that genes are responsible for the manifestation of the characteristics. Let's say, for example, the height of an individual, the complexion of an individual, or the color of hair, the color of eyes. So all those things. So there would be a set of genes which will be responsible for internal manifestation of the characteristics. So such set of genes refers to genotype of an individual. Okay, that is in simple manner to say software. Like how software would be there, software will not be visible, but hardware is visible. Okay, so in the similar fashion, there would be certain genes and which will be responsible for physical appearance of the characteristics. Let's say height, genes responsible for height. So there can be genotype as well as phenotype. So genes that are responsible for physical expression or manifestation of characteristics, that is nothing but phenotype. Okay, so this is the software and this is the hardware. Okay, so likewise genotype and phenotype would be there. So in genetic engineering, what we'll be doing, in genetic engineering we will be altering the genotype, so as to modify the phenotype. Are you getting that? So we will just alter the software so that hardware will be modified. So output would be modified likewise. So that would be thing that we'll be doing here. Okay, so here we are saying that genes are there, and an organism will have a lot of genes. So if we try to have all that complete set of genes of an organism, so the complete set of organisms genes is nothing but genome. Okay, so genome is another technical word that you have to understand. So when we talk from the perspective of genes, an organism will have several genes. So if we try to pick, like for example, genes responsible for height, genes responsible for eye color. So if we map all those things at one place, so we will get complete set of genes of that particular organism. So here genome is nothing but the complete set of the genes of a organism. Okay, so when you understand the genome, then you will understand that what kind of genes are present. Okay, so that is one thing. And if you look from genome perspective, there is a process called genome sequencing. Genome sequencing, through which you can make up, or you can bring all the genome at one place. So genome sequencing refers to mapping of all the genes of an individual, so that we can prepare a map of genetic map of an organism. Are you getting that? Genome sequencing is a process of mapping of the genes of an organism so as to prepare a genome. Is that clear? So genes, then genome, and so as to make the genome, we'll go for genome sequencing. So genome sequencing is that process, okay, mapping of the genes. Is that clear? Okay, so then here we are talking from the perspective of genes, and we have said that they are the carriers, or they store the genetic information. We have said likewise. So what would be the functions of these genes? So usually genes will contain the genetic information, and this genetic information would be manifested in different forms. So certain genes will code for certain proteins, likewise, or certain genes will code for certain things. Okay, so there is a process which, from which, like, genes would be transferred into some manifesting components, like let's say proteins. Okay, so the process by which our genes would be converted, genes means genes contains DNA. So the process by which the DNA or genetic material is converted into some of the useful forms like proteins. So that process of conversion of, generally to say, DNA, the process of conversion of DNA or genetic material into some useful forms. So I am taking an example of proteins. Some useful forms like proteins, that process is generally known as genetic expression. Genetic expression or gene expression. That means genes would be expressed into some useful forms. Are you getting that? That means every gene will have specific function. Genes responsible for making one particular protein, genes responsible for giving height, genes responsible for giving, like let's say, color of the hair, likewise. So every genes will have its function. So in this process is known as gene expression or genetic expression. Okay, so if we talk from the perspective of technical words, this genetic expression is also known by one theory called central dogma of molecular biology. Central dogma of molecular biology. So what do you mean by central dogma of molecular biology? Means it is the doctrine which specifies how genes would be converted into some useful forms. So that's why we are calling it a central dogma. Dogma means it's a kind of doctrine. So central dogma of molecular biology. At the molecule level, what will happen? So that is one thing. So wherever you encounter the word, so this is nothing but gene expression or genetic expression. Genetic expression, again, it is nothing but it is the process of conversion of the genes or genetic material or DNA into some useful products or useful forms like proteins. Okay, so if we talk from the perspective of this gene expression or genetic expression, gene expression is again will consist of some steps. Okay, so if we talk from this gene expression or genetic expression, it usually consists of some steps. Like, let's say, two important steps is nothing but transcription, and other thing is nothing but translation. Transcription and translation are the two important steps in the process of gene expression or in the process of genetic expression. Okay, so what do you mean by transcription? Okay, so usually we have the genetic information in the form of DNA. Okay, so we will have DNA. Okay, so this DNA would be converted into, this DNA would be generally converted into mRNA, messenger RNA, mRNA. The process of conversion of DNA into mRNA or the process of synthesis of mRNA from DNA is known as transcription. Transcription, this is the process, and this process would be helped by an enzyme called transcriptase. Transcriptase is the enzyme which helps in the process of conversion of the information that is stored in the form of DNA to RNA. So transcriptase is the enzyme. Okay, and then what will happen then? This information which has been transcripted into mRNA would be further converted into certain proteins. So this process of conversion of the information which is there in the form of mRNA to proteins is known as translation. Okay, translation. So here in the process of this translation, we will have role of RNA called, like, we will have tRNA, so which will help in the process of this. Okay, so DNA to mRNA and mRNA to proteins. Okay, so this is the two important stages of gene expression: transcription and translation. No, it is an RNA, type of RNA, transfer RNA. Okay, mRNA, tRNA, rRNA, we'll have different types of RNAs. Okay, ribonucleic acids. In ribonucleic acids, we will have different types of RNA. So mRNA means messenger RNA, rRNA means like a ribosomal RNA, tRNA means transfer RNA, like this. Okay, so that would be different types of RNA. Okay, so here during this process, sometimes what may happen means we can also convert the information that is present in the form of mRNA to DNA. Okay, DNA means deoxyribonucleic acid. So the process of conversion of the information that is present in the form of mRNA into DNA is known as reverse transcription. Reverse transcription. So think of some of the virus. So some of the viruses are made up of RNA. RNA is the genetic material among them. So if we cannot directly read that and we cannot code that. So what we will do in such cases, we will try to convert that RNA first. Not audible? Okay. So sometimes what we will do, we will try to convert the information, genetic information of that particular virus which is present in the RNA form to DNA, so that we can read it and then we will use it for further process. Okay, so that process of conversion of the genetic information that is present in the form of RNA back to DNA is nothing but reverse transcription. Okay, so and during this process there would be an enzyme which accentuates or helps this process. So that enzyme would be known as reverse transcriptase. Reverse transcriptase. Okay, so this is one another process. Okay, and there is another process called, here we have a copy of DNA and we can make another copy of the DNA by one method called replication. Replication. So this process is known as DNA replication. Transcription, translation, reverse transcription and replication. So replication means making exact or identical copy of the same. Okay, so sometimes DNA would be made into several copies of same composition so as to read in a better fashion. So DNA replication means making identical copies of the DNA. So that is one thing. So these are some of the important aspects that you have to know, because when we talk about in further aspects, there you will get some of the terms like helicase or like such kind of things. Like for example, the enzyme that helps in the process of this DNA replication is nothing but helicase. Helicase enzyme. So I hope basics, you are all aware of basics. So can you see transcriptase, reverse transcriptase, helicase. So enzymes will end with this terminology. So that would be the terminology that we will use. Case, like can you see at the end S? So that is one thing. So helicase is the enzyme that helps in the process of DNA replication. DNA is generally made up of polynucleotide chain. DNA is nothing but it's a chain of polynucleotides. Okay, so again nucleotides are made up of what? Can you able to recollect? Nucleotides are made up of what? Nucleotide is made up of generally no, not chromosome. Okay, or can you able to recollect the structure of the DNA? Double helical structure. Okay, so what does it consist of? So usually it consists of some things like one pentose sugar. Can you able to recollect? One pentose sugar, then one phosphate group composition of DNA. So then it will have some nitrogen bases. Can you able to recollect now? So what are the different types of nitrogen bases that the DNA will have? Two types. Can you able to remember? That is purines and pyrimidines. Okay, so what types of purines we will have? Adenine and guanine. So we will have adenine and guanine. These are nothing but two types of purines. Okay, so we are talking about DNA. So adenine and guanine. And what about pyrimidines? Thymine and cytosine. Okay, so these are the two types of pyrimidines we'll have in DNA. Okay, so generally DNA is made up of these things. So there, nucleotide is nothing but this structure. This entire structure is made up known as nucleotide. Okay, so this nitrogen bases, phosphate bases, pentose sugar, everything would be composed of one nucleotide. So DNA would be made up of such polynucleotide chains, or it's a chain of polynucleotides. Okay, likewise. So remember, that is one another important aspect that you have to remember. So here, why we are talking about them means, if we talk about this structure of the DNA. So it would be double helical structure. Okay. So when we talk from the perspective of this nitrogen bases would be arranged in a specific sequence. Specific sequence means based upon the composition of the genetic composition. So usually if these are complementary pairs, there are complementary pairs that always adenine pairs with the thymine and guanine pairs with cytosine. So this is the pairing mechanism. So complementary pairs we will call that. That means if on one strand of the DNA, if we have adenine, so on other strand we will have thymine. So this is complementary. So there should not be any changes. If there is there, then that is known as a defect. Okay. So in the similar fashion, if guanine is there, then we will get cytosine. So likewise, we will have the structure of DNA would be made up of. So in genetic engineering, what we'll be doing, we'll be trying to alter this sequence of the nitrogen bases. When we alter this, obviously structure will change. As structure will change, its function also gets modified.